outer layer called as plasma membrane. So he only discovered the plasma membrane. His name was Theodore. Yes, okay. This is Matthias and this is Theodore. And let us see what is the cell theory is all about. And we know all the organisms are composed of cells and their products only. And all the cells give rise for the pre-existing and pre-existing cells. And then it is show similarity in what? Chemical composition and also metabolic activities. And it is a structural and a functional unit of all living organisms. Okay. Now let us see the basic structural outline of a cell. The first one is nucleus. What is a nucleus? Central part of the function. Yeah, and it considered to be a brain of the cell. Okay, nucleus nothing but the brain of the cell, and which is in spherical shape, and it's surrounded by a membrane called as cytoplasm. Cytoplasm, and the nucleus is composed of chromosomes. What are chromosomes? Genetic material. And then another one, the cytoplasm. Cytoplasm, it is a semi-fluid matrix that occupies the volume of the cell. So majority volume yaar occupied pannirukkiற cell? Yes, cytoplasm, and it is mainly composed of water with free floating molecules, in which takes place all the gaseous exchange and also elimination waste and hereditary mechanisms, etc. And the next layer is outer membrane. Outer membrane, it is the outermost layer of a cell which gives protection to the cell and prevents from further injury, and also controls the exchange of ions and all molecules and other components in and out of the cell. Okay, now let us see size and shape. Okay, size ஒரு செல்லும் ஒரு size, அப்புறம் ஷேப்பும் அதே மாதிரி different shapes. Okay, give some example for cell. பாடியில் இருக்க எத்தனை செல் இருக்கு? Yes, everything. Okay, RBCs, WBCs, stem cells, blood cells, muscle cells, and also intestinal cells. Okay, and as I said, all the cells size endless variation sizes and lifespan and cellular activities. Lifespan ஒரு செல்லுக்கு ஒரு lifespan இருக்கும், இல்ல எல்லாருக்கும் சேம் கிடையாது lifespan. And then example smallest cell பேர் என்னன்னா mycoplasma, or otherwise known as PPLO. அப்படினா pleuropneumonia like organisms. And the organism mycoplasm is the smallest cell, and which is approximately 3 to 5, what is that? Yes, meter in size. And then ostrich the largest cell irukku, that is about 170 to 135 millimeter, and humans length 90 to 100 centimeter. And various cells shapes various shapes and all there, maybe polygonal, disc, and then cuboid or irregular. There are two types of cells. Cells ரெண்டு type பிரிச்சிருக்காங்க. ஒன்னு prokaryotic, இன்னொன்னு eukaryotic. Yes, okay. Now we'll see what are prokaryotics. Prokaryotics ko eukaryotics ஒரு difference என்ன? Cell membrane. Yes, cell membrane வேற? Yes, main nucleus தான். Prokaryotics there will be no nucleus, but whereas the eukaryotics they have nucleus. Occurrence எப்படினா all the kingdom under Monera comes under this prokaryotics, and these are the simplest and the most common type of or among prokaryotes. And then they are represented by examples bacteria, cyanobacteria, mycoplasma or PPLO. Mycoplasma, what I said previous, smallest. Yes, okay. Bacteria size approximately 0.3 to 1.3 μm, and then shape we know various shapes. What are the shapes in bacteria? Yes, spherical rod. Yes, okay. Components of a bacterial cell. It is composed of genetic materials, and then envelope, cell envelope, cytoplasm, nucleus. All these are idamloda parts. Okay. Cytoplasm na enna paatha நம்ம முன்னாடி? Cytoplasm na cytoplasm covering, thick coating illa, it's not a covering. இப்போ எல்லா பார்ட்ஸும் அவங்கவங்க பிளேஸ்ல இருக்கு. ஓகே, இப்போ எக்ஸ்ட்ராவா இருக்கு, வெளியே இருக்குல, டென்ஸா இருக்கு, எல்லா பார்ட்ஸும் அதுதான் மேக்ஸிமம் வால்யூமாவை கவர் பண்ணுது. தட் இஸ் சைட்டோபிளாசம் கவர்ஸ் த மேக்ஸிமம் வால்யூம் ஆஃப் அ செல். ஓகே, பார்க்கணுமா இங்கே? ஓகே, எஸ், இல்லை இல்லை, அது ஃபுல்லாக சைட்டோபிளாசம் தான். ஓகே, பார்த்துமா? ஓகே, Now let us see the cell envelope. Cell envelope அதோட modification. Cell envelope தான் it is the outermost covering of a bacterial cell, and it protects what, like mechanical and from injuries, and there are 3 layers. ஒன்னு வந்து glycocalyx, cell wall and a plasma membrane. Glycocalyx na it is a mucilaginous sheath. அப்படினா எப்படி இருக்கு mucilaginous na திக்கா மியூக்கஸ் எப்படி இருக்கும் அந்த மாதிரி இருக்கும். ஓகே, இட் இஸ் த outermost layer yellow இது outermost layer, and with this thickness among different bacteria, and it helps in resisting phagocytosis. What is phagocytosis? Yes, ingesting and engulfing the what? Engulfing what? Food. Okay, phagocyte, appo it helps in that. And then cell wall glycocalyx ku below irukra layer தான், that is cell wall, which is made up of what? Peptidoglycan. It's variously seen in cyanobacteria, and then it is a rigid and a solid covering that gives a shape and a strong structure to a bacterial cell. Appo which gives shape to a cell na? Yes, and plasma membrane is the innermost of the cell envelope. Envelope oda inner plasma membrane da. It is semi permeable. Abdina enna semi permeable na? Semi permeable. ஒரு semi permeable எல்லாமே வந்து போக முடியாது. semi permeable na transparent na எல்லாரும் போயிடலாம் இல்லை, பட் செமி பெர்மியல்னா சில கொஞ்சம் குறிப்பிட்ட சப்ஸ்டன்சஸ் மட்டும்தான் போகும். ஸோ அது என்ன ஆகும்னா ரெஸ்பான்சிபிள் ஃபார் த இன்ட்ராக்ஷன் ஃபார் த செல்ஸ் வித் அவுட் சைட் என்வாயர்மெண்ட், அண்ட் ஆல்சோ ஹெல்ப்ஸ் இன் எக்ஸ்சேஞ்ச் ஆஃப் ஸ்பெசிஃபிக் மெட்டீரியல்ஸ் தான். எல்லாமே வந்து எல்லா மெட்டீரியல்ஸும் கான் கோ இன்சைட் பிகாஸ் ஆஃப் திஸ் பிளாஸ்மா மெம்ப்ரேன். அண்ட் த நெக்ஸ்ட் ஒன் இஸ் ஜெனட்டிக் மெட்டீரியல். ஜெனட்டிக் மெட்டீரியல்னா gene, DNA, okay, and எதுக்கு வந்து இந்த நியூக்ளியாய்டு வந்து represents as a gene genetic material. In case of prokaryotes வந்து that is naked. Prokaryotes is naked and not enveloped by a nuclear membrane, okay, and many bacteria contain a small circular bacteria la DNA is in the form of plastids, okay, circular DNA known as plasmid. Another is membrane structure. What is that means? It is prokaryotic cells வந்து they lack a complex membrane bound organelles, and some special membrane structures are found in them like mesosomes and chromatophores. Okay, next we will see about mesosomes. Okay, mesosomes என்ன முன்னாடி plasma membrane என்னன்னு பார்த்தோம். நம்ம plasma membrane எதுக்கு கீழ வருது? Of yes, cell wall, அதாவது cell envelope பார்த்தோம். அதோட எக்ஸ்டெண்ட் பண்ணுறது தான் இந்த மீசோசோம்ஸ். என்ன ஆகும்னா இந்த ஃபார்ம் ஆஃப் வெசிக்கிள்ஸ் அண்ட் டியூபியூல்ஸாக இருப்பாங்க அண்ட் லாமிலே. ஸோ அதோட எக்ஸ்டென்ஷன் தான் இட்ஸ் நத்திங் பட் மீசோசோம்ஸ், அண்ட் தே ஆர் ஈக்குவல் டு மைட்டோகாண்ட்ரியா இன் யூகேரியோட்டிக்ஸ், அண்ட் தேர் ஸ்ட்ரக்சர்ஸ் வந்து இட் இஸ் ஏரோபிக் செல்லுலர் ரெஸ்பிரேஷன் இன் ப்ரோகேரியோட்டிக்ஸ். அண்ட் அவங்கவுங்க ஃபங்க்ஷன்ஸ் என்ன பண்ணால் தே ஹெல்ப் இன் ரெஸ்பிரேஷன் அண்ட் ஆல்சோ இன் செல்லுலர் செக்ரீஷன்ஸ், and increase the enzymatic content and surface of area of the plasma membrane, and also helps in the formation of cell wall. என்ன ஆச்சு? அதுக்கு அப்புறம் helps in the replication of DNA. Okay, next one is chromatophores. What is chromatophores? Chromatophores adla இந்த wordல என்ன தெரியுது? Yes, chromosomes. And what are they na? They are the photosynthetic forms which helps in which possess a photosynthetic pigments. Chloroplast na chromosomes illa, chlorophyll. Okay, and mean panna chromosomes illa. And these pigments are light reflecting. And what is flagella? Flagella, pili, avangala yaru? Yes, external projection used for what? Yes, okay, but flagella வந்து bacteria can be எல்லா பாக்டீரியாவுக்கும் flagella இருக்கணும்னு அவசியம் இல்லை. they can be motile or non motile also, and the motile வந்து எப்படி இருக்கும்? the flagella வந்து it has a like appendages extending from the cell wall called a flagella. அப்போ flagella extends from what? From where? Yes, and it is classified according to the number of number and arrangement of the flagellum in them. Each flagellum is about 1 to 7 meter long and covered by a protein coat. And okay, these are the different parts in a flagellum. Flagellum 3 parts. One is the filament, hook and a basal part. Filament, it is the longest portion in the flagellum and which extends to the surface of the outside of the cell, to the outside, and is made up of protein called flagellin. Okay, and hook it is made up of protein subunits. Ivanga protein called flagellin. These are made up of subunits of proteins. And then basal body, it is a complex part of flagellum. Complex part na? Yes, complex, yes. Okay, next is pili and fimbriae. Sorry, pili na? Example enna kudunga pili vechirukka idhula. Paramecium, covered fully. Where? Anything? Okay, pili na eppadi irukum? Full projection, flagella madri illama. Yes. Okay, these are elongated tubular structures made up of special proteins called pillin, and fimbriae are very small bristle-like fibers coming out of the cell. When pillin is made up of, pili is made up of pillin, and whereas this fimbriae is made up of what? Fimbriae is made up of like fibers, okay? And then function forming the conjugation tube during transfer of genetic materials between the donor and the recipient cell, and which helps to, this fimbriae helps to attach to the solid surfaces. Okay. Next we will see about ribosomes and inclusion bodies. Okay. Cytoplasm enna na sonna? Yes, semi-fluid. Okay, semi-fluid. In the ribosomes, okay, cytoplasm எப்பவுமே ஒரு cytoplasm என்னது டென்ஸா இருக்கதான cytoplasm சொன்னேன். இந்த cytoplasm எதனால கிரானுலரா இருக்கு? கிரானுலர்னா ஸ்மாலா ஸ்கேட்டர்டா இருக்கும். எதனாலனா due to the presence of ribosomes and inclusions body இருக்கறனால தான் this cytoplasm is like granular in structure. And ribosomes na யாருன்னு பாப்போம். It acts as a site for protein synthesis. அப்ப protein synthesisலாம் எதுல நடக்குதுன்னா ribosomesல இல்ல. And then they are very small but are complex both in structure and chemical composition. And they are about 15 to 20 nanometer in size. Ribosomes it has two subunits. One is smaller subunit and larger subunit. That is smaller na it is 30 years, larger na it is 50 years. Okay. Ribosomes they occur only in helical groups. Helical groups na eppadi? Helical shape eppadi irukum? Double yes, correct. Okay, அப்படிதான் helical groups இருப்பாங்க. DNA helical structure எப்படி இருக்கும்? And they are called as what? Polysomes or polyribosomes. And then in each polysomes there are 4 to 8 ribosomes attached to a single strand of mRNA. Okay, and the next is inclusion bodies. Inclusion bodies na, okay, what are inclusion bodies? Inclusion bodies என்ன பண்ணுவாங்கன்னா, these are the reserve materials stored in the cytoplasm. இந்த இடத்துல தான் they will store what? Reserve materials. Okay, this inclusion bodies, and they are non-living structure present in the cytoplasm and not bonded to any of the membrane system, and they either be life free in the cytoplasm and may covered be with 2 to 4 nanometer thick and non-protein membrane. Okay. Okay, and the next is eukaryotic cell. Eukaryotic cell na, it has well differentiated nucleus and a with a nuclear envelope and several membrane bound organelles is called as eukaryotic cells. And all the eukaryotic cells are not identical except for what? Monorems eukaryotic organization seen all in protists and then plants, fungi and animals. Avanga the cell is larger than of yes, prokaryotic cell. General structure they process a variety of locomotory. அங்க locomotion க்கு என்னலாம் இருக்கு? flagella, pili, fimbriae. But inge vandu there are variety of locomotory and cytoskeletal structures. And cell wall is a special membrane being present in plants, fungi and some protists only. And they have a large vacuole and plastids. Plastids na enna sonna? Yes. Genetic DNA la. Yes. Okay, which is absent in animal cell, and while animal cell possesses centrioles which is absent in plant cells. Okay, plant cell ku animal cell ku enna difference? Enna inga irukum anga illa, edu inga irukum anga illa? Chlorophyll எங்க இங்க இருக்குது? அது chlorophyll என்னது? green pigment. cell wall இருக்காது animalக்கு. இங்க plant அப்புறம் animalsக்கு வந்து centrioles இருக்கும், இவங்களுக்கு centrioles இருக்காது. இதுதான் difference. okay, this is centrioles. ஓகே, நேற்று வார்ம் பிளட்டட் கோல்ட் பிளட்டட்லாம் பார்த்துட்டு வந்தீங்களா? வார்ம் பிளட்டட் என்னது சொல்லிட்டேன் நினைத்தேன் நான். டார்சல் வென்ட்ரல். டார்சல்னா பேக், நீ மாற்றி ஃப்ரண்ட் சொல்லிட்ட. ஓகே, டார்சல்னா பேக், வென்ட்ரல்னா ஃப்ரண்ட். okay, now let us see about the cell membrane. Cell membrane vandu they are covered by a thin elastic transport semi permeable and a regenerative membrane called as what? Cell membrane or a plasma membrane, which we saw earlier in what? Earlier in eukaryotic cells. Okay, this is eukaryotic. In prokaryotic cells, and this membrane separates the internal environment of the cell from the external environment, and also helps in what? Regulating the entrance and exit of the molecules in and out of the cell. So plasma membrane da ellathukku responsible, ulla out and in. And then in 1950, with the advancement of the electron microscope, what they did was they found the detailed structure of the membrane was studied especially on human red blood cells in 1950. okay, இப்போ வந்து நம்ம வந்து ஸ்ட்ரக்சர்ஸ் பார்ப்போம். ப்ரோகேரியாட் யூகேரியாட்டோட செல்லோட ஸ்ட்ரக்சர்ஸ். ஃபர்ஸ்ட் வந்து என்னன்னா, the cell membrane வந்து இஸ் கம்போஸ்ட் ஆஃப் லிப்பிட்ஸ் அண்ட் விச் இஸ் அ ஃபார்ம் ஆஃப் அ பை லேயர். அவங்க லிப்பிட்ஸே வந்து பை லேயராக இருப்பாங்க வித் ப்ரோட்டீன். அவங்க கூட ப்ரோட்டீன் மிக்ஸ் ஆகிருக்கும். ஸோ தட் இஸ் ப்ரோட்டீன் மாலிக்யூல்ஸ் ஆர் எம்பெடட் அட் த பிளேசஸ், and it also possesses proteins and carbohydrates also. Lipid is of two types inge. Okay, lipids na enna papa. First வந்து lipid molecules are what are they na? They are amphipathic in nature and arranged within the membrane by the help of two types of enzyme. ஒன்னு வந்து polar hydrophilic, another வந்து polar non-polar hydrophobic. Philic phobic அப்படினா? Yes, phobic na? Okay. இவங்க வந்து என்னன்னா எங்க எண்ட்ல இருப்பாங்கன்னா, வாட்டர் எங்க இருக்கோ அந்த எண்ட்ல இவங்க இருப்பாங்க. this polar hydrophilic end, and faces towards the outer side of the cell with interact with the aqueous environment to the on the both side. Whereas non-polar na anga water, they won't interact with water environment. That is the region is water repelling. Both the ends of the faces faces each other that occur towards the center of the cell membrane. And the protein of the lipid vandu they vary from cell to cell, all types of plasma membrane. And the form cholesterol vandu ratio la vandu 20 to 32%, and mainly of phosphoglycerides and phospholipids, which is ratio enga irukudhu. The cell membrane possesses protein. The lipid molecules are amphipathic in nature அப்படினா they are liquid in nature. lipids எப்படி இருக்கும்? lipids வேண்டாம், எண்ணெய் எப்படி இருக்கும் அந்த மாதிரி. ஓகே, வழ குழக்குறேன். ஓகே, and then okay, proteins பத்தி பார்ப்போம். proteins வந்து depending on the ease of extraction, the erythrocytes வந்து has approximately 52% of protein and other 40 வந்து lipids. proteins வந்து ரெண்டு டைப்பா கிளாஸிஃபை பண்ணிருக்காங்க. ஒன்னு வந்து integral and peripheral. integralனா வந்து stronger, பெரிஃபரல்னா வந்து வீக்கர் அசோசியேஷன். ஓகே, அது கார்போஹைட்ரேட்ஸ் அடுத்து வந்து என்ன கார்போஹைட்ரேட்ஸ் காம்போசிஷன் வந்து 1 to 5%. அண்ட் தீஸ் ஆர் அசோசியேட்டட் வித் பாஸ்போலிபிட்ஸ் ஆர் பெரிஃபர். இவங்க வந்து they are linked with proteins to form a glycolipids and glycoproteins respectively. Okay, and the next what we gonna see is fluid mosaic model. Okay, இது யார் டிஸ்கவர் பண்ணது? Any idea? Any scientist name? Guess. Okay, it was given by Singer and Nicholson in 1972. Singer, S-I-N-G-E-R. Okay, Singer in 1972. அவங்க என்ன பண்ணாங்க, இந்த what this model state is the bilipid layer. Okay, நம்ம என்ன படிச்சோம், plasma membrane is composed of lipids, proteins and carbohydrates. இவங்க என்ன சொல்றாங்கன்னா இந்த lipid லேயரும் protein லேயரும் வந்து ஒரு mosaic மாதிரி ஃபார்ம் ஆயிருக்கான். mosaicனா டைல்ஸ் எப்படி இருக்கும் வீட்ல, டைல்ஸ் அந்த மாதிரி mosaic மாதிரி ஃபார்ம் ஆயிருக்கு அப்படின்னு சொல்றாங்க. அதுக்கப்புறம் ஓகே, the ability of the protein வந்து எப்படி மூவ் பண்ணுது அப்படின்னு சொல்றாங்கன்னா the fluidity of the membrane சொல்றாங்க. அவ்வளோ ஃப்ளூயிடா இருக்கனால அது நல்லா மூவ் ஆகுது அப்படின்னு சொல்றாங்க. அது வந்து ரெண்டு டிரான்ஸ்போர்ட் இருக்கு, ஒன்று வந்து ஆக்டிவ் and other one is passive. ஆக்டிவ் இதில் ஆக்டிவ் இல்லையோ? ஓகே, ஓகே, active transport, passive transport la the model வந்து they have carried out. Active transport na movement against the concentration gradient using energy, that is ATP. But passive வந்து without energy. Active la என்ன நடக்குதுனா lower to higher concentration போகுது. Okay, lower to higher. Okay, இது பாப்போம். இதுல என்ன ஆகுது அப்படினா okay, இந்த plasma membrane வந்து என்ன எப்படி இருக்கும் நான் சொன்னேன், plasma membrane எப்படி செமி பெர்மியபிள். செமி பெர்மியபிள்னால எல்லாமே போக முடியாது. ஸோ இந்த மாலிக்யூல்ஸ் அயான் வந்து உள்ளே போயிட்டு இப்படி வரணும் வெளியே, உடனே அவங்களால போக முடியாது. ஸோ அதனால they have a carrier protein. So with the help of the carrier protein, this molecule ions gets inside it and forms here, and which ATP is converted into ADP plus phosphate. What is ATP full form? Adenosine triphosphate. அப்போ D வந்துச்சுன்னா it is diphosphate. So in the presence of carrier protein, okay, so அவங்க ஈஸியா permeable ஆயிட்டு உள்ள உட்கார்ந்து உள்ள they'll form here. Another வந்து passively வந்து it is without energy, no energy is used here, and is of three types. ஒன்னு வந்து osmosis, another simple diffusion and facilitated diffusion. Osmosis இங்க கொடுக்கல, I'll tell what is that. Okay, okay, osmosis னா என்னது? Osmosis? Osmosis na என்னது? படிக்கலையா? Osmosis மேலே ட்ராக் பண்ணுறதா? ஓகே, என்னன்னா இப்போ okay, osmosis na water molecules passes from the higher concentration to lower concentration. Osmosis nale water, okay, what does it do? Yes, okay. And where is this simple okay, diffusion na? Any idea diffusion? Yes, diffusion na கலக்கிறது. Simple diffusion na என்ன பண்ணா neutral molecules வந்து they'll pass to this plasma membrane through a channel protein. Anga it is carrier, osmosis la it is in osmosis higher to lower edu? Yes, water, but simple la it is through neutral molecules passes through the plasma membrane in the presence of a channel protein, and facilitated na in the presence of again carrier protein here. Okay, sorry, facilitated diffusion na channel protein, carrier protein na it is simple. Okay, sorry, first I'll make it clear. Osmosis na movement process of waters to enter into the plasma membrane from higher to lower concentration. Simple abdina, it is from carrier protein. Neutral molecules வந்து enters into the plasma membrane in the presence of with the help of carrier protein. இப்போ வந்து facilitated na with the channel protein like ions and all. Okay. Active na it is then passive means without energy. Okay, let us see the functions of cell membrane. Cell membrane oda functions la enna va irukum? Cell membrane, yes, protection from external injury and avanga semi permeable irukanaala allows only specific substances to enter into the cell, and then it also have a carrier proteins for the transport and also contains enzymes which performs various reactions in the surfaces, like example ATPase and phosphatase. Okay, next we'll see is cell wall. What is cell wall? Yes, outer covering, and it was first discovered by Robert Hooke. And cell wall is absent in animals but only present in plant cells. Okay, it is differentiated into three parts. One வந்து middle lamellae, primary, secondary. Middle lamellae na eppadi irukum? Irukko. Okay, middle na center la irukku. It is made up of calcium and magnesium pectate. Okay. இப்போ வந்து சிமெண்டோட வேலை என்னவா இருக்கும்? While yes, அதே தான் இந்த middle lamella பண்ணும். Okay, adjoining the cells, two cells or more cells. Okay, cement மாதிரி அது work. And the primary cell wall அப்படி என்னன்னா ஒரே நிமிஷம். Okay, primary cell wall na middle lamella குள்ள இருக்கிறது வந்து primary cell wall which is young and growing cell. அந்த primary cell க்கு உள்ளார வளரதான் that is secondary cell wall. Okay, lamella குள்ள இருக்கிறது primary குள்ளால இருக்கிறது secondary. Okay. And the middle lamella is made up of what? Primary cell wall. Magnesium and calcium pectate. Okay. This cell walls provides a definite shape and structure to a cell, and then it also helps to cell interaction. Okay, cell interact பண்றதுக்கு it will be helpful. And also prevents damage and injury from external. Okay, another is endomembrane system. Okay, what are the organs which constitute an endomembrane system? Any ideas? It is here, right? Endoplasmic reticulum, cell wall. These are this consists of a nuclear envelope, and then endoplasmic reticulum, Golgi Apparatus, all are suspended in the cytoplasm, and they are considered together as an endomembrane system because their functions are coordinated with each other, and in spite of this that each membrane organelle is distinct in terms of structure and functioning. Okay, now let us see about endoplasmic reticulum. What is endoplasmic reticulum? Endoplasmic reticulum என்ன ஃபங்க்ஷன் பண்ணுது? அது ஃபங்க்ஷன் சரி, endoplasmic reticulum என்னது படிச்சிருப்பீங்கல்ல? membrane bound algae. ஓகே, அதோட ஃபங்க்ஷன் என்னவா இருக்கும்? லைக் ஃபங்க்ஷன்ஸ் main they contain various enzymes to process the metabolic activities. Okay, endoplasmic reticulum நிறைய enzymes இருக்கு to conduct what? Metabolic activities. Yes, okay, now let us see about endoplasmic reticulum, and it is a complicated system which is of membranous channel and flattened vesicles. It is relieved from an electron microscope only of eukaryotic cells, and that the presence of a network of reticulum or tiny tubular that has been scattered in the cytoplasm. And then there are two compartments, one is luminal, another is extraluminal. Luminal na inside the reticulum, endoplasmic reticulum, it is it is present near the cytoplasm, that is outside. And there are two types of reticulums. What are they? Smooth and rough. Okay, any differences between them? Smooth and rough? Surface smooth ah irukum. Okay, எதனால என்ன ஒரு ரீசன் இருக்கும்? எதனால? இவங்க எதுக்கு ஸ்மூத்தாக இருக்காங்கன்னா they don't possess or bear a granular ribosomes. granular scattered-ஆக இருக்கனால தான் அங்கே இருக்காமல் இருக்கனால they are smooth. but roughல வந்து they have a scattered what is that ribosome, so they are rough. this is a basic difference between them. smooth endoplasmic reticulum la தான் various synthesis நடக்கும் like lipid synthesis, protein synthesis and all. okay, but whereas rough la don't take any synthesis there. that's all. என்ன ஃபங்க்ஷன் சொன்னேன் உங்களுக்கு? metabolic activities like எதனால metabolic activities அங்க நடக்குதா? அப்புறம் they contain enzymes so that they are very useful in metabolic activities. and they also help in rapid transport of the channels. okay, transport irukum, they are helpful in that. okay, and the next is Golgi Apparatus. Golgi Apparatus na Golgi Apparatus, any functions or any what is that? okay, we'll see. It is discovered by Camillo Golgi, so the name was named after him only, and it is a major complex protoplasmic structure being made up of very flat and a disc shaped sacs of cisternae and in diameter. அவங்களுக்கு வந்து ரெண்டு phases இருக்கு, two distinct phases. one is cis phase, that is helps in the formation of the phase, and other is trans phase, which is a mature phase. எல்லாம் வளர்ந்துட்ட பிறகு that phase is called as what? Trans phase. trans phase. okay, function வந்து these are involved in the formation of lysosomes. lysosomes na enna? yes, correct. okay, correct. and they are appo ivanga da they help in formation of lysosomes and also help in perform the function of packaging materials. or material package ஆகிறதுக்கு they are very helpful. helpful in the production of complex carbohydrates. okay, any example for complex carbohydrates? glycogen, glucose. okay, oh en kaadula vidala na vera edhu nenachiten. and helps in the formation of cell wall also. okay, now let's see about lysosomes. okay, this Golgi apparatus produce the lysosomes. okay, and these are otherwise known as what you said? yes, and now let us see the functions of them. they digest the food contents in the body, and then they also perform the extracellular digestion also, and the old and useless organelles of this cells are present in this, and they also functioning in the cell division. cell division now? okay, now next what we gonna see is vacuoles. okay, these are the large membrane sac found in the cytoplasm. where it is found? the blue one. okay, and these store substances not only essential for the useful of the cell, but like water, sap, these are the extra things which not romba useful illa, so they also serve, I mean what, preserve this also. and the plant vacuoles contain not only that is vice versa. okay, contains not only water, sugar, salts, but also contains pigments and toxic molecules, and occupies up to 90% of the volume of the cell. so 90% is occupied by what? okay, cell wall la majority of volume irukkiradhu okay, and the vacuole is bounded by a single membrane structure called as what? Tonoplast. the outer layer of the vacuole is called as the tonoplast, and then which facilitates the transport of material and some ions against the concentration gradient inside the vacuole. and then two types of vacuole, contract vacuoles and foot vacuoles. contract vacuoles என்னவா இருக்கும்? yes, osmoregulation, that is osmotic pressure idu pandrathu. foot vacuole na? yes. okay, the next one is mitochondria. mitochondria na? Yes, okay. These are the membrane bound cell organelles which is essential for the aerobic respiration in all the eukaryotic cells, and these are called as the powerhouse of the cell as it produces the cellular energy in the form of ATP. Okay, now let us see some functions. What does it do? Any functions of mitochondria? Powerhouse of the cell nale enna function? Yes, control. There is a control, and also provide the intermediate for the synthesis of several biochemicals like pyrimidines and alkaloids, and also has a enzyme synthesis for the fatty acids. Last one next one and the regulation in cellular metabolism, and also apoptosis. Apoptosis na? Apoptosis. Apoptosis. Cell death. Okay, next one the plastids. Plastids na nama munnadi enna paathom? Yes, okay. These are membrane helipal. Plastids have their own genetic material, that is DNA. Due to their large size, enna aguna, they can see under the microscope itself, not ultra microscope but normal microscope itself, and then they are found in plant cells and euglenoids except in some protists only. Like example, euglena, and these are the parts that, yes, correct. These are the parts of the plastids, like inner membrane. Inner membrane edu? Innermost da inner membrane. One is inner membrane and intermembrane space. Appo inner membrane ko inner membrane ko oka space dan, that is intermembrane space, outer membrane, and then stroma, thylakoid and lamella. Okay, and the next one what we gonna see is plastids. Plastids வந்து அவங்க based upon their color, the plastids வந்து vary ஆகும். First one என்னது? Etioplast. இவங்க வந்து they are crystalline. Crystalline எப்படி இருக்கும்? Crystalline. Sugar மாதிரி. Okay, and they are precursors of chlorophyll. Precursors na? Chlorophyll. Precursors of chlorophyll. Chlorophyll ku munnadi, that is precursors. Okay, before. And chromoplast வந்து they are in red or yellowish color, which useful in the pigment synthesis and pigment like chlorophyll synthesis and storage. Chloroplast na green color, adukaprom useful in photosynthesis and also lipid synthesis. Lycoplast na color என்னவா இருக்கும்? They are colorless. Lycoplast na colorless, useful in the synthesis of fatty acids and then amino acids. Amyloplast na storage of starch. Starch na carbohydrates. So they contains carbohydrates, so they are helpful in the storage of starch and sugar. Elaioplast na they are stores lipids and oils. Proteinoplast na they are the site for the enzyme activity. Okay, now let us see some functions of chloroplast. Chloroplast or functions என்னவா இருக்கும்? Yes, that is help in photosynthesis. வேற என்ன இதெல்லாம் இருக்கும்? Release of oxygen in photosynthesis, that is consumption of carbon dioxide, whereas release in oxygen in the photosynthesis plants. And also change the chromoplast in order to provide color to many flowers and fruit, and helps in storing fat and lipids, and also used in the transduction of energies. Okay, next what we gonna see is ribosomes. Ribosomes munnadi paathame. Okay, correct. Okay, ribosomes. Sorry, okay. These are small subspherical granular organelles, and they do not bound to any plasma membrane, any membrane. That ribosomes were first observed by who? George Palade, and as the dense particles under the electron microscope, and hence it was also called as Palade particles. They are mainly composed of what? Ribonucleoproteins, that is RNA plus proteins, and it is also known as protein factories, as they are primarily involved in the synthesis of proteins. So protein synthesis வந்து நடக்கிற site வந்து ribosomes. Yes, Okay. And we know it is prokaryotes, it is 70 S, and whereas in eukaryotes it is 80, and both 70 and 80 S contains two subunits, that is smaller and larger. Smaller enadhu? 30. Bigger, I mean larger? Yes, okay. Now let us see some differences between 70S and 80S. Presence வந்து இவங்க எங்க presence in prokaryotics and other cells like autonomous organelles, mitochondria, plastids. இங்க வந்து only in eukaryotic cells, and வந்து இங்க எங்க synthesized in the cytoplasm, but whereas 80S வந்து it is synthesized in nucleus. Okay, size வந்து they are about 20 nanometer, இவங்க வந்து about 25 to 30 nanometer, and type வந்து they are free floating in cytoplasm. இங்க வந்து they are free floating தான் in where it will float? In nucleus. Okay. And then subunits வந்து 50 and 30, இங்க வந்து 60 and 40 only. And then inhibit, what they do is they inhibit the protein synthesis of 70S ribosomes is inhibited by a cycloheximide. இங்க வந்து chloramphenicol. Any uses of chloramphenicol? Chloramphenicol என்னது? It is used in ear infection and all, chloramphenicol. Okay. Okay, now cytoskeleton. Cytoskeleton na? Okay, நமக்கு வந்து வெளியனா skeleton அந்த இது cytona, it is involved in cell. Okay, and the network of interconnected proteinaceous filaments and tubulase which extends from the nucleus to the plasma membrane. So nucleus ல இருந்து இருக்க plasma membrane, that is known as cytoskeleton. Function வந்து maintain shape and cell, and then its extension, and also gives mechanical support and what? Motility. Motility na? Motility. Okay, now it's cilia and flagella. What is cilia and flagella?
External projections.
That's correct. Okay, external projections we saw in earlier slides. Both are almost identical in structure and but differ somewhat in length only. And then cilia வந்து small structures working as oars, while flagella வந்து longer in size. This is cilia and flagella, which is both are responsible for movement of the cell only. It is based upon organism. ஒரு organisms it differs. Okay, but both function in the same though. Okay, and the next is centrosomes and centrioles. Centrioles எங்க இருக்கும், இங்க இருக்காது? சொல்லு சரி, plant cell இருக்காது, animal cell இருக்கும் centrioles. Okay, and the centrosome is an organelle that has two cylindrical structures called as centrioles. So centriomes oda structure, two types of cylindrical structures that is called as centrioles, and they are surrounded by a amorphous pericentriolar materials, and they both lie இப்படி தான் perpendicular. Perpendicular எப்படி?
90 degree.
And some of the functions we'll see. What are the functions na? These form a spindle fibers and move to the poles at the time of the cell division, and then helps in the formation of cilia and flagella of the cells also. So formation of cilia and flagella helpful, I mean yaar help பண்ணுவதுக்கு?
Centriole.
Yes, okay. Now there is nucleus. Nucleus na முன்னாடி என்ன பார்த்தோம்? Yeah, that is a brain of a cell மாதிரி வச்சுக்கலாம். Okay, it is specialized and a principal organelle of the cell which contains all the genetic information and controls the essential process related to metabolism and heredity. எல்லாமே இங்கே தான் நடக்கும். I mean இதான் கண்ட்ரோல் பண்ணும். And it was discovered by who? Robert Brown in 1831. Functions வந்து control cellular functions also, and then synthesis of structural proteins and stores the genetic information, and also induces genetic variation. Okay, next is chromosome. Chromosome na? X-shaped genetic material. Yes. Okay, these are meant for equal distribution of genetic material, and a single எப்படி இருக்கும்? They have a 2 meter long thread of DNA distributed among 46 chromosomes we know in humans, and then chromatin is composed of chromatin na? Yeah, இப்போ வந்து இத ஒரு full image வந்து நம்ம chromosome சொல்றோம். இதோட half the chromatin. Okay, and then chromatin is composed of DNA and some basic proteins called as histones, and some non-histones proteins and RNA are also present in histones thavara. RNA and non-histones also present in chromatin, and it is composed of a primary constriction or this is centromer, and then on the other sides of the disc shape, on the sides of which the disc shaped structures are present known as kinetochores. Okay, now let us see the இப்போ இந்த சென்ட்ரியோமர் எங்கே பொசிஷனில் இருக்குது, இந்த குரோமோசோமோட பேர் வேறு வேறு மாறும் பேஸ்ட் அப்பான் த பிளேஸ் வேர் இட் பிளேஸ்டு. ஃபஸ்ட்டு வந்து டெலோசென்ட்ரிக் அப்படின்னா பாதி ஹாஃப் இல்லாமல் ஒரே ஒரு ஹாஃபில் மட்டும் அந்த சென்ட்ரோசோம் இருந்துச்சுனாக்கா இட் இஸ் டெலோசென்ட்ரிக். ஆக்ரோசென்ட்ரிக்னா ஒரு பாடி வந்து பெருசாகவும் இன்னொரு பாடி ஆர்ம் வந்து ஷார்ட்டாக இருந்தால் ரொம்ப சின்னதாக இருந்தால் இட் இஸ் கால்ட் ஆக்ரோ. கொஞ்சம் இருந்தால் சப் மெட்டாசென்ட்ரிக். வென் இட்ஸ் ஈக்குவல்னா இட் இஸ் மெட்டாசென்ட்ரிக், தட் இஸ் பி ஆர்ம் அண்ட் yes. Ana metacentric it will be equal, but Q arm is called as long and P வந்து short. Okay. Okay. இவங்களோட functions பார்ப்போம். This control cellular differentiation and also contains all the hereditary information and then forms a link between the parents and the offspring. So chromosomes only are responsible for that all now. And then it introduce variations through process of crossing over and also control cell metabolism. And the next what we gonna see is microbes. Microbes na enadhu? Microbody, sorry. Microbodies na? Okay, yes, small, minute da. These are composed of enzymes, and these microbes are where microbodies enga irukum? Cytoplasm. Okay, composed of cytoplasm. Engela irukun paatanaka in liver, in kidney, and then protozoa and yeast and other types of cells also. Okay, and 3A 3 part da, that is one the crystalline